Today, we're going to be talking about Mendelian genetics. You'll see why it's called Mendelian in a little bit. So, before we start, we should review some important vocab that we've learned so far this semester. One very important word that we've been talking about nonstop are genes, which are sections of our DNA that code for a specific trait. We also have gametes. We talked about those last unit as sex cells. Those are the egg cell or the sperm cell, egg for female, sperm for male. And then we have a zygote, which is the fertilized egg. This is occurring after the sperm and the egg cell meet, but before it becomes an actual baby. So here's why we call it Mendelian genetics. Gregor Mendel, who is pictured on this slide, is known as the father of genetics. Genetics is the study of heredity, or the study of how traits are passed on. Gregor Mendel was an Austrian monk who began to make observations about pea plants. One of the things that he noticed was that there was patterns in these pea plants between different generations. So if both parents were yellow, then the pea plant would be yellow. But if the pea plant had a green parent and a yellow parent, then it'd be green. So those were some of the observations he made. He also took a look at shell texture, so if they were wrinkly or smooth, and he began to notice all of these different things. And from this, he was able to discover genetics and how traits are passed on. So one of the things that he discovered was alleles. Alleles are different forms of traits. So you can have an allele for blue eyes, brown eyes, green eyes, etc. So all of these eye colors are a different allele. It's just a different version of the trait. There are two types of alleles. There are dominant alleles, which we represent with capital letters. So in this case, big A. And then there are recessive alleles. And then these are represented by our lowercase letters, so little a. There's also two very, very important words that we're going to use this unit and next unit, and they are genotype, which is the actual alleles inherited. So your actual genes, genotypes are the genes, they're the genetic makeup. So if you have like big A, little a, big A, little a, and we're going to talk about what that means in a little bit. And then there's something called a phenotype. Phenotype is physical. It's the physical characteristics that you see when you look at an organism. So genotype is the actual genetic makeup. Phenotype is the physical trait. Two people can have the same appearance, but have different genetics. So two people can have the same phenotype, but actually have a different genotype. And we're going to talk about what this is in a second. But for example, this would be like if you had the trait for big B versus someone else having big B, little b. So what does that even mean? So we have three different words that we use when we talk about genetic crosses. Genetic crosses are how your traits line up. So there you have the option of being heterozygous, which is different genes where you carry one dominant trait and one recessive allele. So you have both the dominant allele and the recessive allele. So this would be like big B, big C, big T, big Y, little y. You also have something called homozygous dominant. These are gene pairs that carry two dominant traits. So big B, big C, big T, big Y, big Y. So homozygous dominant is two of the dominant traits. Then we have homozygous recessive. These are gene pairs that carry two of the recessive traits. Homozygous recessive is much harder to have. That's why it's more rare for people to have blue eyes and blonde hair and why it's more rare for someone to have straight hair. And this is where you would have little b, little b, little c, little c, little t, little t, little y, little y. The beginnings of these words, so hetero means different, and the prefix HOM in homozygous means same. HOM means same, hetero means different. Because in heterozygous you have one of each allele, but in homozygous you have two of the same alleles. So we have something called the law of dominance, which we started to kind of talk about in class last week, which is where a dominant or a strong allele will show itself over a weak or recessive allele. So in the example that we'll talk about, if you have dark hair, that is a dominant trait. So that would be big B. If you have the recessive trait, little b would code for the recessive trait of blonde hair. If you have brown hair, you can either have big B, or big B, little b. Because that dominant trait expresses itself in big B, little b, you would see the darker hair color. If you have blonde hair, you would have little b, little b. All right, so for example, if you have a parent that has brown hair and a parent that has blonde hair, so let's say mom has brown hair, dad has blonde hair, and the child has brown hair, that child got a dominant allele from mom, so it has big B, and then it got the recessive allele from dad, so it would be little b, and that child would be a mixture of both of those traits, but he or she would show the brown hair gene because brown hair is dominant in the world over blonde hair. Heterozygous children, so big B, little b, of different traits can often, that's why you might see like two brown haired parents have a blonde haired child because they both have that recessive trait. Another example would be that brown eyes are dominant over blue eyes. Brown eyes can be represented by big B, or big B, little b, but blue eyes would be represented by little b. So again, those big letters are dominant. So if you have one of those dominant traits, you're going to show the dominant trait. We use something called Punnett squares to figure all of this out, which is what we're going to be doing this unit. Punnett squares are diagrams that show the probability or chances of inheriting traits from parents. So, for example, here we have on this screen two parents on the side over on the left side of the square. You'll see that there is a big B, little b. The big B, which is red, is dominant. The little b is blue and it's recessive. We also have the other parent on the top, which is where we have the big B, little b. So this is where you have two brown-haired parents having a blonde-haired child, because when those genes meet in that first top left box, you have the big B and the big B meeting together, because you can use your fingers and make them meet through the Punnett square. You can have another option in the top right corner of the big B and the little b meeting. That's where you would have the heterozygous offspring. On the bottom left corner, you would have that little b meeting that top big B. So you have another heterozygous child. And then you have the little b and little b meeting in the bottom right box. So this is where you would get that blonde-haired child from. So when you look at a Punnett square, it's telling you chances. So This Punnett square right here is telling us that the chance of these two examples, these two parents, having a brown-haired child and a completely homozygous dominant child is 25% because it's 1 out of 4 of the boxes that we have big B, big B. If we were asking what are the chances of having a heterozygous or a mixed child, you would have a 50% chance because 2 out of the 4 boxes have big B, little b. And then if we asked what are the chances of you having a homozygous recessive child, that would be 1 out of 4 chances, which is 25%. So this is why 2 brown-haired parents can sometimes have a blonde-haired child. So this is an example of Gregor Mendel's experiments that he looked at with the pea plants. So green is dominant over yellow, which is why it's represented by the big G. So on this example, we are crossing a heterozygous pea plant on the left with a homozygous recessive pea plant on the top. The genotype of the heterozygous plant would be green. The phenotype is a physical characteristic, so the one on the left would be green. The one on the top would have a phenotype of yellow. And then when we cross them, we get in the top left box, we get big G, little g. And then in the top right box, we get big G, little g. So that means those top 2 boxes would be the 50% chance of a heterozygous offspring. If we take a look at the bottom boxes, we get 2 chances of having little g, which means that we have a 50% chance of getting a homozygous recessive offspring. So when we put this all together, the offspring have a 50% chance of having a green phenotype and a 50% chance of having a yellow phenotype. That's it for the notes today. So make sure you followed along in your guided notes, and then we're going to start talking about what all of this means in class.